Recently, you learned all about the different types of microbes, eukaryotes, prokaryotes, viruses, and you learned about how they reproduce, how they survive, and how they function. And one of the key commonalities that they all have is a genome that allows them to do this. So what is a genome? A genome is all of the genetic information that's found inside of a cell or a virus. This could include DNA for cells, or DNA and RNA for viruses, and it may include DNA that's found in chromosomes or extrachromosomal DNA. Let's draw all of this out together so that you can get the terminology straight in your mind. The genome. We'll look at eukaryotic genomes, prokaryotic genomes, and even viruses have genomes. For cells, the genome is always made out of double-stranded DNA. In eukaryotes, the majority of the genome is going to be found in chromosomes contained inside of the nucleus. And as you may recall, those chromosomes tend to be linear, discrete pieces of DNA. And in prokaryotes, they also have a chromosome that tends to be circular, contained in the nucleoid region rather than in the nucleus. But this is still chromosome DNA. Both eukaryotes and prokaryotes may have what's called extrachromosomal DNA as part of their genomes. This includes plasmid DNA, found primarily in prokaryotes. These are small circles of DNA that are outside of the chromosome, and they contain genes that can be very useful to the cell but aren't essential. The essential genes that are required for the cell to survive are stored in the chromosome. An example of extrachromosomal DNA in eukaryotic cells is found inside of mitochondria and chloroplasts. As you recall, these evolved from a prokaryote, which had its own genome at one point in time, and some of that genome was maintained, and mitochondria and chloroplasts still have their own DNA, even though they are now part of the eukaryotic cell. Right, so as you can see, chromosomal DNA is inside the chromosome. Extrachromosomal DNA is outside of the chromosome. What about viruses? Viruses we're going to get into a lot more detail with later, but for now, let's just say that the viral genome can be either RNA or DNA. And actually, some DNA viruses can take up permanent residence inside of a eukaryotic or prokaryotic cell and can become part of the extrachromosomal DNA that make up the genome of that cell. So let me just review this quickly. A genome is all the genetic information in a cell or a virus, including the chromosomal and extrachromosomal DNA or RNA. Here we're looking at an example of some prokaryotic cells, Bacillus cereus and Bacillus anthracis. You can see the chromosome here. And then there's also shown here some plasmids. The plasmids that are present in Bacillus anthracis actually encode toxins, which are what make Bacillus anthracis a pathogen, whereas Bacillus cereus doesn't cause disease at all. And here we have a eukaryotic chromosome, and if we were to unwind that chromosome and look at it, we'll see the genes that are running along the length. Shown in this red-brown color are all of these genes. And these genes are called coding DNA because they encode a protein. However, there's also DNA that is non-coding DNA, shown in blue here. And the non-coding DNA is often involved in regulation of gene expression or other functions. Not all of the functions of non-coding DNA are well understood, and originally it was thought that it was just junk DNA filling in between the genes. But the more we learn about it, the more we realize that it does actually have some functions. This is a review of the various types of nucleic acid found in genomes. For cells, this is always going to be double-stranded DNA, which can include all of these different types of chromosomal or extrachromosomal DNA. For viruses, this includes DNA or RNA, which may be single-stranded or double-stranded. We'll learn more about that when we get to the lesson on viruses. And this image is just showing the different sizes that genomes can come in. Viruses have some of the smallest genomes, and that's because They rely primarily on the host for a lot of the functions like metabolism and reproduction. And so they don't need those genes. The host stores those genes in their genomes. Viruses only need a few genes that direct the cell in how to make more viruses. And as we move up to bacteria, bacteria have larger genomes than viruses, fungi a little bit larger than that. And then when we get to these multicellular eukaryotes like animals and plants and mammals, these have some of the largest genomes, which kind of makes sense because they have more complexity. They have more different cell types that require more genes to create those phenotypes. And just as a reminder, these are the ones that all have a DNA genome, whereas viruses may have DNA or RNA genomes. So we know that all of this DNA contains these things called genes, and somehow these genes allow the cells to function and survive and reproduce. But how do they do it? Well, the answer lies in the proteins that are encoded by the genes. Each gene encodes a protein, and that protein, when it's made, is going to do all of the work of the cell that allows that cell to function. Let me show you what I mean. Let's take a chromosome, for example. Chromosomes are long, double-stranded pieces of DNA that have many, many genes on them, thousands of genes in each chromosome. Those genes are just discrete sequences that run along the chromosome. For example, let's call this section gene A. And here we have gene B. And here let's draw gene C. Gene A encodes the protein, protein A. Gene B encodes the protein, protein B, and gene C encodes the protein, protein C. In order for the cell to make any of those proteins, it will have to undergo what is called gene expression. Gene expression is where a gene gets turned on, and then that gene is transcribed and translated to create the protein. So gene expression is the same thing as transcription and translation. And when a gene is turned on, we call it expressed. And when a gene is turned off, it is not expressed, meaning it's not being transcribed or translated at that time. So let's follow gene B for a moment. Let's say the cell needs some more of gene B in order to stay alive. Maybe gene B is a transporter for bringing glucose into the cell, and the old transporters got worn out, and you need some more of these transporters to bring glucose in. Of course, the cell can't survive without glucose very well. That's the main energy source for this particular cell. And so it's very important to make more of this protein B. So gene B encodes protein B, and it's going to be transcribed through the process of transcription into gene B mRNA, or we'll just call it mRNA B, right? So this is the DNA gets transcribed into the RNA. And now this RNA will be used as a template for translation in order to build protein B. And now the cell has protein B, so it can continue to survive. In this case, gene B was turned on to make protein B. Another way to talk about the genes that are found in a genome is to talk about the genotype. A genotype is just all of the genes that are present in an organism. And together, all of these genes being turned on or turned off and creating their various protein products are going to create what is called the phenotype. The phenotype is the observable characteristics of that organism, right? All of these proteins are going to create all the things we can see and observe. The color, the shape, the size, the metabolism that it can do. All of these things we've been talking about so far in the class are really the phenotype of these cells. And these phenotypes are due to the collection of proteins that a cell has in it at any one time. And this phenotype is primarily created by the proteins that are present in a cell at any one particular time, and those proteins are encoded by the genes found in the genotype found in the genome. Here's a great example of genotype and phenotype with a microbe that might be familiar to you from lab. This is Serratia marcescens. Remember, you streaked this out in the T-streak in lab? Serratia marcescens can make either no pigment or pink pigment, depending on the environmental condition, where all of the bacteria grown on these plates have the exact same genotype. However, at 37 degrees, you get phenotype A, where no pink pigment is produced. It's only at this lower temperature of 25 degrees Celsius that Serratia marcescens starts making this pink pigment. And so you have this through differential gene expression. At the 25-degree temperature, the genes needed to make this pigment are expressed. All of this may have you wondering, what is the purpose of making this pigment at a lower temperature? Well, it's not known for sure, but one hypothesis is that when Serratia marcescens is in the soil, this is going to be closer to 25 degrees, and some pigments can have antimicrobial properties, and so maybe this pigment is allowing Serratia marcescens to compete with other microbes in the area by inhibiting their growth. Whereas at 37 degrees, Serratia marcescens may be in the body of a host where that type of competition isn't as necessary. However, this hasn't been proven and is still under study.